So good morning, everybody. It's a pleasure to be here. It's a very, a beautiful view and a very nice group of people to discuss and to present CellVax. I'm Fernando Kreutz. I'm the CEO of the company. I'm also the founder of the company and also the inventor of the technology. So I combine several hats in this. With me is Emerson. Emerson is our CFO and helping us in this path of developing CellVax Therapeutics. So I'm going to tell this story about how this technology was initially discovered, what we've done with the technology, and how we're placing this technology in terms of the market opportunity for this type of therapy. In the first day of the meeting, the first talk actually was quite interesting because we passed through different types of therapies that are being using to treat patients with cancer. And what we saw was, so I am an MD by training and a PhD, and I started to work with immunotherapy in the late 90s. So from there to here, we saw tremendous development. And this is what I'm going to actually present to you guys here is like 20 years of work in terms of conception up to where we are at this point. So it's a really, really fascinating story on this. So we have a small disclaimer here, of course. I'm going to walk through this, but this is basically a review of where we are and what are our next steps. So I'm going to tell and I'm going to describe how we actually reached to a point where we actually ready for a phase 2 trial here, looking at high-risk prostate cancer patients. And our therapy, what it does is actually prevent that those patients go into recurrence. So it's a quite unique mechanism of action, first in class in terms of mechanism of action. And also another thing which is quite unique in our approach is the first in class in terms where we actually are applying this technology. So in a quite unique position for treating patients. So I'm going to start by a very basic discussion. So when we look at how the immune system interact with, between the cells, so we know that's, the mechanism by which whatever is happening inside the cells is actually presented to the immune system is through the MHC. So we basically have MHC class 1, which actually present whatever is happening inside the cell. So every time we have a new antigen or a mutation that is actually translated into a protein, we know that this is going to end up in the MHC class 2. At the same time, so the MHC class 1 will actually interact with the T cells, T cytotoxic cells. On the other hand, we also need the MHC class 2 that is necessary in order to activate the immune system. So we have this two signal component. So we need not only the neoantigens on the cell surface of the cell, but at the same time, I need this second signal in order to activate the immune system. So what we saw early on is that we have what we call the cold tumor situation. So where basically we have the mutations are there because cancer, we need mutations to have cancer. There is no cancer without mutations, so we know that there are mutations. But the problem is that this does not activate the immune system. So the mechanism by which I'm going to describe here is a way to actually activate the immune system and transforming those cold tumors into hot tumors. So, and this is have a very interesting discovery. So I was in the lab doing an experiment. I had 3 extra vials, 3 extra tubes to be tested. And what we do as a scientist, well, let's add another negative control in here and see how that goes. And by my surprise, so what I did is I treated the cells and I was looking for, cell surface changes. I was looking at some of tumor markers, and I put a negative control as MHC class 2 antibody. And by my surprise, what happened was I actually had an expression of MHC class 2 onto those cells. So now I have a epithelial cell, a tumor cell, that is not only presenting MHC class 1, but is now presenting MHC class 2. Oh, that's quite interesting. So what we do is, okay, let's see if this is dose dependent. So let's see if we can, repeat this and see if, increase the dose, increase the response. Let's see if we can do this with different cells. So we've done that, and yes, we can do all that. So we can demonstrate that yes, it's dose dependent, and yes, this happened in several different cell lines. That's very nice. Now, the other question comes, okay, that's nice. You have MHC class II in the cell surface, but are these MHC class II actually presenting antigens or presenting peptides on it? And yes, we are able to demonstrate that not only we have positive MHC class II now on those cells, but this actually are presenting peptides on it, and presenting peptides coming from the media that we actually grow in the cells. Oh, that's quite interesting. So what we can do with that? So then I came up with a proposal, and the proposal was what we call a tumor presenting cell. So what's that? That's actually having a tumor cell that usually does not express MHC class II, but by induction, I have now the cells expressing also MHC class II. And by doing that, what can happen with this cell? So this cell, we actually can be effective in activating the immune system because it's actually it's doing that two-cell function in a single cell. So it's not only talking to the T cytotoxic cell, but also talking to the T helper cell. And we know that we need this in order to activate the immune system. And the immune system works in a very close proximity. So we need to have all this happening at the same time. So this was a model. So this was then a proposal. And our hypothesis was that, yes, those tumor cells can act as activating T helper cells and activating T cytotoxic naive cells. So this would be a perfect candidate for immunotherapy approach. Of course, the other question was, okay, by doing this, I am activating the cells, not only activating lymphocytes, not only against the modified cells, but also to the unmodified tumor cells. And yes, as we was expecting, we are doing that. So now I have a potential therapy in order that can, in one hand, have the expression of different neoantigens on MHC class I, and at the same time, have a secondary signal into the MHC class II. So here is another big challenge that we have in cancer, which is cancer is a very heterogeneous disease. So we know that if we look at tumors, and if you look at side-by-side tumor cells, the cells will be different. The cells will have different mutations. So we need to have a representative sample of this in order to activate the immune system. And more than that, the immune response should not be target to a single protein, to a single epitope. Because when that happens, it's very easy for the tumor to escape that by just down-regulating that specific protein. So with this strategy, we actually are presenting hundreds of different peptides from different neoantigens to a different population of T cells. So this is quite unique in terms of a strategy. So we went further and we went and say, okay, let's design a trial. And let's design a trial in, so usually, and this is something that's I think in our approach, to look at disease that are really important, really relevant in terms of care. So we saw a lot, I think a couple of months back, I saw a presentation that say, well, the last drugs that we actually approved, that the FDA approved, will actually represent only 2% of the cancer population. So we need therapies that actually gonna be applied, to cancers that have a really important play in the overall scenario. So here we have prostate cancer. of course, you guys can understand that this strategy is not only limited to prostate cancer. It could actually be applied to basically any solid tumor that we are able to upregulate the MHC class II pathway. So, of course, prostate cancer, it is a major issue. We know in prostate cancer that we have a window where we basically, after prostatectomy, we don't do anything. We just wait to see what happens with that patient. So if I can design a therapy with low toxicity that has an important response, this will be a great model to be tested. Talking about animal models in this type of therapies are extremely difficult to create, because not only you have to match the tumor, but you have to match the MHC, not only class 1, but class 2. So it is really complex to do this. So we went further and we developed this as a phase 1. So we done this in Brazil, initially as a phase 1 trial. Then we moved this into a phase 2 trial. And we combined data from this study. And we look at this. And so we combine all this, and we create a dossier of this, and we present this to the FDA into a pre-IND meeting before the pandemic. So we get all this data, and we create this, and based on our data, and I'm going to show you the data that we present to the FDA. So basically, what we have is these are a group of patients high risk of recurrence. So we know in prostate cancer that we have 5 levels of risk of those patients having recurrence. This is based on the tumor size, based on the PSA level, based on the Gleason situation. So if I take patients that are in this high or high risk of recurrence, we know that despite the prostatectomy, so despite of having the surgery, those patients will have recurrence of the disease. So what we did, we designed this to be used in high-risk group of patients. So indeed, actually, when we designed the trial, this was a phase 1. So as you guys know, usually when you do a phase 1 trial, we have some bias, and we have a bias, of course, against the vaccine arm, because, higher risk group of patients, if you don't randomize the trial, you're going to end up in the treated group. So indeed, we have, a worse population in the treated group than the control. But what we demonstrated is, so what we have here is actually, we follow this patient. So we have some patients, so this was a trial that was done, that we initiated several years back. So we have, in some patients, we actually have 10 years follow-up of those patients. And what we demonstrated is that by doing this treatment, we gain 27 months free of recurrence. So this is to say more than 2 years that the patient do not need to do any other treatment. So this is impact not only in cost, but also impact in terms of the patient quality of life. Because, first line of therapy for prostate cancer is hormone therapy, ADT. Well, ADT have several significant side effects. So if I can prevent that, or if I can delay that, that is very, very significant in terms of impact for this population of patients. So as you can see, our hazard rate is very significant. So this is not a therapy that is, has a little effect on the overall patient progression. It actually have a very significant response. So although this study was not powered by that, what we demonstrate is that we had in the control group, we have up to 36% recurrence rate against 12% on the treated group. And remember, we have actually a worse group in the vaccinated arm. So this is very interesting in terms of results. Mortality in prostate cancer, because we have several different lines of therapy, is very hard to measure, and usually because of new therapies it comes, it's hard to do that. But even that, we actually demonstrate that there was a trend in terms of mortality reduction as well. So those are very interesting data. So now it comes a second point, which for this therapy is quite unique. So the first scenario is unique in terms of mechanism of action. So we are focused on the tumors themselves. We are not focused on the infiltrating lymphocytes. We're focused on the tumors themselves. So the other fact is, when you think about immunotherapy, what is the best place to test this type of therapy is when I have a healthy patient and when I have minimal residual disease. And what we done was, with this protocol, we actually done this right after the prostatectomy. So we had a chance to evaluate this therapy at a very interesting and a very nice opportunity, which is patients are right after prostatectomy are healthy, and the patients are if they have residual disease, it is a minimal residual disease. So this is the perfect time for actually doing this therapy. The other aspect of this, and this was present in our discussion with the FDA, was can we do this can we repeat this trial now in the US in this group of patients? And those are one of those very emotional moments when you do this pre-IND meeting. You know, you go with them and they say, okay, well, let's think about this. And, 5 minutes later, they come back and say, okay, you can go ahead and do that. So we are very pleased to be able to test this and design the trial that we're doing this in this very nice side of population, not only in terms of the patient opportunity to be using this treatment, but again, as a market opportunity, fantastic, because you don't have that many drugs in the same space. Because usually what happens, the drugs are pushed back into the metastatic patients. So this is quite unique in terms of and in here, we have this several other drugs that are being used and approved for prostate cancer. Now, imagine this, and the data that we generated showing this, that by doing that therapy there, I reduce by half the recurrence of the disease. And if I do that, I also reduce by half the need of all those other drugs. So this not only have an impact on the patient quality of life, but also has a tremendous economic impact. But overall, all this that we spend in prostate cancer, it can reach up to $20 billion in the US alone. So imagine I have a therapy that actually can reduce this by half. So challenge here is, so we moved so CellVax was incorporated here in the US in 2020. We move all the IP and all the clinical data into this. And challenge number one, okay, we need to manufacture this product here in the US. Challenge on cell therapy products, when you talk about investing and talk about cell-based products, you say, oh my God, this is gonna cost you a fortune. Well, interesting enough, we probably have the lowest cost, manufactured cost of a cell-based product. We're talking about $20,000 per patient cost of manufacturing this. And this starts with a tumor fragment that goes into the lab. We expand those cells, and then we modify the cells. We do not introduce new genes. We just activate genes that are already there. So it's a very straightforward strategy in terms of manufacture.
Just we believe in terms of scalability, we can reduce those costs per patient you might have.
So we actually contract a very interesting CMO in California, and they have top-notch facility. We've been very active with them in terms of tech transfer. We've been successfully growing tumors with them for the next 3 months now. So we're developing a bunch of new assays that are necessary for the CMC portion of our IND submission. We have a very straightforward pathway in terms of regulatory pathway on this. Interesting enough, we proposed this protocol for 50 urology centers in the US. Out of those 50, we got 43 that were interested in participating in the trial. We are able to select 20 sites that are going to be participating in this trial. So this is the design of the trial. This is a very sophisticated trial. We have several different aspects of this following. So this is targeted for just after the prostatectomy in this population of patients. This is our flow in terms of what are the next steps. So we expect that until the end of the year, we'll have all the data that we need to submit the IND, and we hopefully will have first patient in either in the end of this year or in the beginning of next year. We designed this trial as have interim analysis in several points, so we may have opportunities to evaluate this in several points during this development. Interim analysis, looking at different populations of patients, high risk, low risk. Of course, in this pathway, secondary indications of this, it's a completely open, not only for different tumors, but also by different regions. So we know in some regions we have, of course, in China, prostate cancer is not a major cancer type, but we look at gastric cancer there, which is a huge impact there. So just to finalize our team, one of our members and shareholders is Robert Sims. Robert was actually one of the oncologists involved in Provenge. As you guys know, Provenge was one of the first immunotherapies strategies approved for prostate cancer more than 10 years back. And Bob was really involved into this. And when he saw this technology, he was really on board on this. We have a very neat clinical advisor board, from different specialists in the US. And the challenge now is we actually raised $5+ million to go up to the IND submission. And now we're looking for $20.5 million to do this phase 2 trial in prostate cancer. So we are looking at this point to partners for that, to investors for that, and also looking at potential extension this for secondary indications and for secondary markets. Thank you very much.
Unfortunately, we don't have any time for questions, but we can obviously discuss during lunch later today.
Absolutely. Thank you.